Welcome to Bio 1104, and this is the third in the series of nutrition, growth and control of microbes. There's some references there for you, but there's also those electronic resources that I have on Study Desk for you, plus there's a number of different resources online, such as TODA's online microbiology. We're going to be we looked at some physical aspects of antimicrobial use or effectiveness in the last one, but in this one we're going to be looking at the mechanisms of action of antibiotics and also some of the physical sterilisation means as well. So we're going to look specifically at heat, low temperature, high pressure, radiation filtration and high osmotic pressure. Okay, so these are all things that can influence the growth of microorganisms. And what I want you to be able to get out of this is the mechanism of action of antibiotics and also some of the mechanisms that we're going to be talking about for controlling microbial populations as well, together with their limitations, but you also need to know how they work, okay, to actually appreciate what their limitations could be. We'll start off with antibiotics, and these are, you typically think about antibiotics as being something that you get from the pharmacy, but antibiotics are produced by microbes. And you can see that quite easily, because if you have a lawn culture of something and you put an antibiotic on it, like those discs, then you get a clearing zone where the organisms are susceptible to that antibiotic. And that's one of the tests that you can use if you are taking a particular type of antibiotic for an infection and that infection doesn't clear up. They take a swab of that infective material, culture it, and then they do a series of tests for it. So you use those little discs with antibiotics impregnated on it, and so you can see what's resistant and what's susceptible. Okay? And how that came about was, it was in 1928, Alexander Fleming left a window open and he had some Staphylococcus aureus growing on a plate, and he had a mold spore that came in and alighted on that plate. You know how many mold spores there are in the air? So in the environment, you got some beautiful cultures last week with that. So what happened was, mold sometimes takes a little bit longer to grow. So with that growth, you need to give it long enough to grow, and then that mold will actually send out metabolites, enzymes, through the media to affect the growth of things around it. Okay? And that's exactly what Fleming saw. He saw a mold and a clearing zone around it where Staphylococcus aureus wasn't growing. So what he did, he put it to one side, and it was only in 1939 that at the Australian, what's his name, Florey, Alexander Florey, not sure what his first name was, Howard Florey, and he worked with Ernst Chain, and they took that discovery from Alexander Fleming and put it to work. So they started to make a lot more penicillin. Now, penicillin is a really good antibiotic, and if you were being injured in World War II, it would be good to have an antibiotic. Yes? And that's exactly what happened. In the early 1940s, the antibiotic penicillin was being used to give to people who are injured. However, Chain and Florey couldn't produce enough. So what happened was some of the penicillin, when it goes into the body, it comes out of the body unchanged via the kidneys. You seeing where I'm going with this? Okay, so what happened is people realised that penicillin was in very short supply, and that's number one. Number two is penicillin could be recovered from urine 3 to 4 hours after it was administered. So the urine was collected and the antibiotic was isolated and reused. So that's the story of penicillin. Okay. So most of these antibiotics are produced by things like streptomycin, Streptomyces fungi and penicillin. And they produce a number of different types of antibiotics. You've probably heard of tetracycline, clavulanic acid, those sorts of things. These are produced by bacteria, or microorganisms, sorry. And Bacillus species will produce, you can get polymyxin and things like that. And for them to produce that, it's really important because they can then influence their environment. So if you don't think about antibiotic production that we can use as humans, if you think about them in their environment, if they can discourage the growth of competing microorganisms, then they've got more access to nutrients so they can grow. So it's a really good protective mechanism for them. But when we use antibiotics, we look at a number of things. We have to look at how toxic they are. So if you've got something that is an antibiotic, for example, or a chemical that is toxic to a cell, is it more toxic and you don't get too much benefit from it? So it's that toxicity benefit trade-off that you have to think of. So selective toxicity, you want really to be affecting the bacteria that might be causing, or the microorganism that is causing a disease, rather than the cell, the host organism. Okay? Then you have to think about spectrum of activity. Does it kill everything? Is it a broad spectrum, or does it have a very narrow spectrum? Can it only kill one particular species, for example? And then you have to think about the modes of action, because how it works may have an effect on the host tissue as well. So if we start with selective toxicity, you think about the property to kill or inhibit microorganisms without damaging host cells. That's important. When you get selective toxicity, when you're measuring it, you can go, it's a scale of 1 to 8. So if you've got a rating of 1, that means that it's less effective and it's more toxic. Say a rating of 8, it's more effective and less toxic. Okay, so there are a range of values for that. You can have highly toxic antibiotics. You certainly wouldn't be ingesting them. They're the ones for external application. So I thought I had an example of one. Anyway. So obviously external application rather than internal, that's important. So that mode of administration, so is it superficial or deep? Then you have to think about the max, how much of a dose can I give? With some antibiotics or some chemicals, you also have to think about the age of a person. So if this chemical is distributed through the water, through the fluids in your body, as you age, that fluid volume decreases. So you certainly wouldn't give the same concentration or dose to a young person to an old person, sorry, that you would give to a younger person. Because what would happen? In that older person with less fluid, with the same dose, it would become concentrated, more concentrated, and then it could be toxic. Okay? Spectrum of activity, what does it act against? So if you've got a broad spectrum like tetracyclines, they're effective against a wide range of microorganisms, gram-positive and gram-negative. Okay? But a narrow spectrum, such as penicillin G, now penicillin has got a number of different types. Penicillin G is just one of those types. It's effective against a small subset of the gram-positive bacteria. Now, how do they work? How they work is grouped into a number of different sets. So you've got the cell wall. If you interfere with the cell wall synthesis, then you wreck the integrity of the cell completely, don't you? And it can't survive, it can't replicate. If you disrupt the cell membrane, once again, you get that change in osmotic pressure and the cells will lyse eventually. If you inhibit protein synthesis, then the cell can't make those proteins or enzymes that it needs to metabolize. So that will stop it replicating. If you inhibit nucleic acid synthesis, so if you stop their genetic material being replicated, then the whole cell can't replicate. And if you have an anti-metabolite in there as well, that just means that you put in something extra, and so when the cell is producing its or replicating its DNA, the anti-metabolite goes in there as well and stops the proper replication of DNA. So it's a nonsense DNA, so therefore it can't replicate. So if we go number one, cell wall synthesis, so penicillin stops that occurring, vancomycin. Polymyxin disrupts the cell wall membrane function. The erythromycin, streptomycin, chloramphenicol inhibits protein synthesis. You've got nucleic acid synthesis being inhibited by quinolones and metronidazole, and anti-metabolites such as trimethoprim. And sometimes these anti-metabolites, as an antibiotic, if you have it just by itself, it might be slightly effective, but if you join it, if you have two together, it actually becomes more effective. So you get that synergistic effect as well. So if we look first at the cell wall synthesis, these are the antibiotics are known as the beta-lactams. And that just means that the beta-lactams block the enzyme needed to form the cross-links in the peptidoglycan cell wall. Okay, that's all it does. It just stops an enzyme. So instead of the cell wall being nice and firm and robust, then it's not produced very well at all. And that sounds really good, except with all good bacteria, they can have a lot of genetic variation, and so we've got an increase in cells that they produce an enzyme known as beta-lactamase, which means they break down beta-lactam, so it's ineffective. Okay, so you've heard of microorganisms being resistant to penicillin. These are the ones that produce the beta-lactamase. Okay, and with the pili and the conjugation pili, you can get transfer of those beta-lactamase genes between bacteria as well. And we've got some examples there, so the vancomycin and bacitracin. And it's just, this is the gram negatives, and here we have the gram positives as well. So you will get some cell death, but sometimes you'll get cell survival as well. Okay. This is just basically showing you where the disruption occurs. So if you have polymyxins, which are effective against gram-negative bacteria, they bind to the phospholipids, so you'll get disruption there. If you have binding to the sterols, then, so if you've got a fungal disease, for example, such as amphotericin B, it will bind to the sterols and disrupt the cell membrane. Okay, so it's basically, if they bind, then they disrupt and it just loses all its integrity. Now, protein is used for enzymes and lots of things internal to the cell. And if you wanted to inhibit that in bacteria, say if you've got an infection and you take a particular type of antibiotic, that might affect the bacterial protein synthesis. Okay? It could work on the 30S subunit or the 50S subunit. So streptomycin, tetracycline work on the 30S, and chloramphenicol and erythromycin work on the 50S subunit. Okay? So that sounds terrific. Except remember ribosomes? So remember our mitochondria? So they have the 50S and the 30S subunits as well. So even though we might be inhibiting protein synthesis in the bacteria, we're also inhibiting the action of the mitochondria in our own cells. So sometimes there are unexpected reactions. So nucleic acid synthesis, so it's, we spoke about that, but yeah, basically it's inhibiting the DNA or RNA. If that's not replicated, then the cell can't replicate itself. Acting as an antimetabolite, so it's incorporated into molecules, as I said. So if you've got a drug or a substrate, it will compete for a particular site, and then you don't get an end product. And if that end product is a protein, it won't be made, or if it's DNA or RNA, it won't be made, or it will be nonsensical. Okay, so you stop that being produced. So this is just quite a nice summary of the modes of action, just showing you the same thing again, just different way. Sometimes you might recognise or realise what's happening with one image rather than another image. But we've got the inhibitors of cell wall peptidoglycan, protein synthesis, metabolic inhibitors, and nucleic acid synthesis inhibitors, and the mRNA synthesis being inhibited as well. Now, physical ways of decreasing microbial population. So, as I said before, you can have heat. So, you know, in the prac, we use heat, so with flaming loops, etc., to get rid of microorganisms. Okay, so we can use that in a larger scale, so we can use dry heat or moist heat, or we could even use pasteurization. So short, not as high a temperature, but for shorter times. And that's not sterilizing, that pasteurization, it's decreasing the population. Okay. You could use low temperature, so refrigeration or freezing, that might help to decrease microbial populations. Might be drying. So have you seen very much microbial growth on jerky? Because it's had most of the moisture taken out. So for microorganisms to grow, they need available water. Okay? High pressure, that is one way. High pressure is sometimes combined with heat in an autoclave to decrease or to sterilize substances. You can use radiation as well. Radiation is quite nice to use for sterilization, but it only sterilizes the surface. Okay, it doesn't actually penetrate. So you have to know what you need to be, where the population is that you need to control in order to say this is the best method. Filtration. Now in the prac we filtered some water, and basically we have the filter paper and there's certain size of pores, so it will capture some of the microorganisms. If you wanted to filter sterilize, you would use, instead of a 0.45 micron filter, you would use a 0.22 micron filter. And you can do that by putting something in a syringe, putting on a sterile filter, and just pushing down the plunger, and what comes out will be sterile. And obviously it'll have to be collected in a sterile container, and everything done aseptically. Then we have osmotic pressure. So back to food: jerky, jam, jellies. Jerky has got a lot of salt, it's decreased moisture. Jellies, lots of sugar. Okay, so you increase that osmotic pressure. Unfortunately, if you do that, then you might decrease the population, one type of population, say the bacterial population, but you might allow a lot of yeasts and molds to grow. So now with heat, it is very good to use with materials that aren't affected by heat. If you're going to use heat and the substance that you're using the heat on denatures, then it's not much point using heat. You have to find some other way of decreasing microbial load. It will allow rapid penetration, so you get that penetration which is needed, and it will denature proteins of the bacteria. And it will also be, with not just the proteins, it will be denaturing the lipids in the cell membranes. Okay, so there's another couple of hits to that. Now there's a couple of definitions with this. So the thermal death point is the lowest temperature at which all bacteria in a liquid culture will be killed in 10 minutes. Okay, so once again, you've got the temperature, but you've got a specific time frame as well. If you've got that time frame, then you can compare something that we do in Toowoomba to something that's done in Melbourne or London, for example, okay, because you've got that protocol. Then the thermal death time is the length of time required to kill all bacteria in a liquid culture at a given temperature. Okay, so it's just changed slightly. So that's the thermal death time. And we saw a graph earlier, so in the last lecture, about the decimal reduction time. So the DRT is the length of time in which 90% of microorganisms will be killed at a given temperature. 90%, i.e. 1 log decrease. Yes, do you remember that image, that graph? Now, heat can be given as in a dry environment, and it can be used to sterilize metal or glassware, oils, powders, things that you don't want to be moist. Okay, you want them to be kept dry. So it could be 180 degrees for an hour or 160 degrees for 2 hours. So they're the typical times that you need for that type of sterilization. And you can have open flames used in the laboratory, which we've used. You might have incineration as well. Now, what is one of the problems that you could see with using a Bunsen burner? So an open flame in a laboratory. Sorry? Yes, so there's workplace health and safety. Probably not, because you're using aseptic techniques and you've got all of your PPE on. But sometimes if you're not careful enough, you might produce aerosols. Okay, so you need to be aware of what you're using. Okay, and that's why there are certain techniques to use when you're flaming a loop, for example, to decrease that incidence of aerosols. Okay. Moist heat is, if you add moisture, then you can get faster penetration. So you need less time with that heat. Okay, and it will also denature enzymes and disrupt membranes. If you think about moist heat, you might think about boiling, and that's fine. Boiling can kill a lot of vegetative cells, but it won't kill endospores. They can survive boiling for quite a number of hours. And it can also inactivate some viruses. Probably more effective is autoclaving. So this is steam under pressure. So you've got temperature, moist heat, and pressure. And that an effective method of moist heat sterilisation, but the steam must come into contact with the substances or the materials to be sterilised. Okay, so if you jam pack everything together and steam can't circulate through, then what's happening in the middle won't be sterilised. And typically you use 15 psi, which is pounds per square inch, and 121 degrees C for 15 minutes. That's the normal. Sometimes that can be varied if some, say, media is sensitive to that temperature for that time, but it will kill vegetative bacteria, endospores, and disrupt that viral DNA or that viral genetic code. So it's quite an effective way of sterilising. And what it looks like is you need a steam supply. Okay, so you've got a chamber here, you've got a wheel here, so on a door. So if you open it up, you'll be looking down that way. And so then you put what you need to be sterilised, and it might be some media that you want to be sterilised, put it in there and then close the door and make sure it's tightly closed and locked. And then you turn on the steam supply, and that steam will circulate around, but it will also circulate through. Okay, so that media has to be in contact with that steam. And when that steam comes in, it will actually displace the air that's already in there. Okay, so it's just basically voiding that air. And the air is just removed. So in that pressure chamber, you will have steam being circulated and an increase in temperature, plus you'll have pressure. Okay, and that is how you sterilise a particular thing with using an autoclave. You've got a lot of pressure gauges, and you can set them so they stay at that particular temperature and pressure for the allotted amount of time. So it's not when you turn it on, it has to be coming up to that temperature and pressure, and that's when the timing starts. Now, it's all well and good if you think about one particular thing. So one bottle of media, for example. But how can you tell if it's effective? So what you can use is some autoclave tape. Now, it goes in and it's kind of clear, but then when it gets up to temperature, there will be black lines start to be produced. That's just when it gets up to temperature. It doesn't tell you how long it stayed at that temperature and pressure. So this is just, it's an indicator of one thing. But what you can also do is put in a vial. This is just a vial of bacteria. Bacteria that can withstand a lot of heat and that forms endospores. So typically you would put one of these in each load, and you'd put it in the middle of the load because remember you need that penetration. And then when everything has, when the cycle is finished, you take this bioindicator out and incubate it. And what happens is if it's got a colour indicator in it, typically, if there's something that, if all of the microorganisms in here have been killed, then it won't change colour. But if there's something growing, it will start to metabolise and ferment, and you get a change of colour to yellow. So you get production of acid. So it's quite easy to see after a bit of, probably about 48 hours, 24, 48 hours, if this autoclave cycle has been successful. And that's why generally you have a label when this was autoclaved and when it was checked. Okay, so you have that quality control. Pasteurisation, we spoke about that with milk. So what does that mean? It is that high pressure, but high temperature, not as high, but higher temperature for particular amounts of time. And typically it will kill the more important food poisoning microorganisms. So things like Salmonella and Mycobacterium will be killed. And then the other bacteria that might be present will actually, their populations will be decreased. Okay? But you know yourself, if something's pasteurised, it doesn't mean it's sterile. If you left your milk for too long, it would start to grow, okay, and produce gas or whatever is in there. Some of the pasteurisation could be ultra-high treatment, high heat treatment as well. And usually we think, well, we'll just pop it in the fridge, nothing will grow in the fridge, or pop it in the freezer, nothing will grow in the freezer. Well, not quite true, because if you have those cyclophiles that are in the food stuff, then their growth might be retarded, but they won't be killed. And sometimes they could start replicating, but very, very slowly. Okay? Even in the freezer, some microbes can keep replicating, albeit very slowly, but they can replicate. So it's not a method of controlling them forever. If you were using low temperatures, it would have to be very, very low temperatures. So if you have, oh no, we don't need to go into that. Listeria, has everyone heard of Listeria? Yeah. Frightening PhD project a number of years ago now looked at Listeria concentrations in soft serve ice cream in France. So soft serve ice cream is kept at quite a cool temperature before it's distributed. Listeria can survive those sorts of temperatures. Okay, this is what I was talking about as well. So some survive those low freezing temperatures. Freezing actually does help with killing off worms or worm parasites in meat, which is good. And some microbes can be stored at very low temperatures. So minus 80, but they have to be stored in glycerol. Okay, so it's a way of keeping them in stasis, basically. So if you want to have a stock culture, you might freeze it in glycerol at -80, and then after a year you might bring it out and you can actually put it in growth medium and it will start to grow again. Okay, so it hasn't been killed, it's just in a stasis. Then we've got desiccation, so that just means that things are dry. So you take out the water, take away the available water for the microorganisms to grow, and then that's a way of decreasing populations or retarding growth. So you've looked at dried fruit. They can stay quite stable, microbiologically speaking, for a while. Not only are they drying, but sometimes they add other types of preservatives such as sulfides. Okay, that will also decrease growth. But if you think about beef jerky, they can stay growth-free for quite some time. Dry surfaces inhibit the growth of a lot of organisms, such as the causative agent of syphilis. And this is environmental conditions affect the efficiency of desiccation. So saliva, pus, feces, and mucus are protective. Okay, so the causative agent of TB can survive in saliva on the sidewalk for many, many weeks. Okay, so saliva, pus, feces, etc., they can protect the organism. Bless you. High pressure will denature proteins in living cells, and obviously if it's just high pressure by itself, endospores won't be affected, and that high pressure might actually kill all the vegetative cells, but be enough to prompt that endospore to germinate. And then if there's another wave of high pressure, then that germinated endospore will be killed as well. So it might, like a two-punch, two-part decrease in population. And high pressure can be used to preserve fruit juices, etc. And that is great because you're not getting the temperature associated with it, so you're not going to get a change in the organoleptic properties of that particular food. Okay, so if you put a particular foodstuff in an autoclave, yes, you'd sterilize it, but it might change the properties so it'd taste revolting. Or you might use just high pressure on that same foodstuff and you keep all the properties, colour, taste, texture of that particular foodstuff. Okay, so it just depends what you're dealing with, what you're wanting to occur. Radiation can also be used if you use wavelengths, lower wavelengths, you can damage DNA and proteins, and it will destroy viruses much better than bacteria. But it has to be direct, and it's only on the surface. So UV light, you will be able to, if you clean the surface and then put on a UV light, it would sterilize the surface. Okay, now this is actually a really nice image. So what you've got is a plate of agar, and you've got Serratia marcescens growing, so this orangey pinky colour, and you've got this part which was irradiated, UV light. So you can see that most of the growth has been inhibited, but some hasn't been. Okay, so it's generally effective, but you have to be aware that sometimes it may not be as effective as sterilisation. But depending on where you're using it. Ionising radiation, you've got a great deal of penetration, but it will destroy DNA. So if you've got lab or medical products, so if you've got metal prostheses, for example, you really want that sterile. So you might use radiation. And that's at Lucas Height, that's where you get a lot of the ionising radiation sterilisation occurring. Now, filtration is one of those things that you used for the water, and this is just an indicator of what could be done with that. But you have to think about that filtration membrane. Okay, so if you've got a 0.22 micron filter, that means that the pore sizes are 0.22 of a micron. That means that viruses will go through, but bacteria won't. Okay, you can get much smaller pore sizes to filter viruses and proteins if that's what you need. Okay, microbes can also be filtered from the air, so we're talking about liquid here, but they can be filtered from the air using HEPA filters as well. Now, this is a large filter, but this is what I was talking about, the syringe filtration. So you've got the syringe filled with a fluid that you want filtered, and this would be a sterile filter, and then this would be all sterile. So on this side wouldn't be sterile, but here would be sterile, okay, because you'd be using aseptic technique. The osmotic pressure that I was talking about, if you destroy the cell membrane or the cell wall, then you get plasmolysis. So instead of the cell being nice and happy in an isotonic solution, then if it's put in a hypertonic solution, then you will get the movement of water out, because if you destroy that plasma membrane. So that is what plasmolysis is. But with a bacteria, that's usually effective, but some molds and yeasts are capable of growing in materials with high osmotic pressure. Okay, back to food. Have you seen a lot of bacteria growing on your jam that you've forgotten about? Or is it mold? Mold or yeast? And that's what it would be, mold or yeast, because they are able to grow in environments of high osmotic pressure, so high salt, high sugar. And this is just another summary page from Black and Black, just going through what we've spoken about. Once again, it's got the action, what the agents actually do, and then how they're used. Okay, it's quite a nice way of looking at it. So we've looked at antibiotic use and how we can control microorganisms by targeting different parts, different metabolism steps, and different antibiotics can have different effects in the cell, and how those different classes of antibiotics actually work. And we've looked at a number of different ways to physically control the populations of different microbes, from sterilisation to a decrease in population. And we've looked at the different modes of action and how efficient they actually are. So this is time for you to test yourself. So beta-lactamases are, and the sterilisation equipment used in a laboratory to generate moist heat is a or an. What do you think? Everyone got it? Ish? Make sense? Yes? Okay. Well, thank you very much, and I will see you next week.